In this video, Dr. Colleen Prell of the University of Texas at Dallas gives a brief overview of how noise, cisplatin, and aminoglycoside antibiotics induce cell death in the inner ear, and how investigational drug agents have protected the inner ear against cell death and hearing loss, preserving auditory function not only in preclinical lab studies completed in rodent models, but also in early-stage clinical trials. Dr. Laprell's presentation was hosted by the Chicago section of the Audio Engineering Society. For more information and events, visit the Chicago section website.
All right. Well, thank you everyone for making time to come and listen tonight. I hope that I'll be able to share some information that will be either interesting or useful in some way. I'll confess I taught an entire class on this topic this semester, so I've tried to take about 30 hours worth of material and boil it down into the next 45 minutes. So I'll do my best not to try and repeat all 30 hours and keep you here until midnight tomorrow night. All right. So before I begin, I just want to first thank the different agencies that have funded our work. Right now we're receiving funding from the Department of Defense for the work that we're doing on noise-induced hearing loss and some of its earliest symptoms and kind of how risk begins, how risk grows. We also have funding from NIH for some of the preclinical work that we're doing, and from 3M for some work with hearing protections. I have had funding from industry previously, Sound Pharmaceuticals and Edison Pharmaceuticals, and I'll talk just a little bit about some of the clinical trial data from the studies that were supported by those groups. I am also a co-inventor on a couple of patents that are owned by the University of Michigan, and those are related to some dietary supplements that we've worked with both in preclinical and clinical trial paradigms. So I want to cover two broad topics tonight. The first one is drugs that can harm your hearing, so that we're all thinking in the same way about some of the ototoxic risks that are out there. And then after we talk about some of these most common drugs that cause negative effects on hearing, we'll shift gears and we'll talk about drugs that have been helpful in protecting hearing. And one of the things that is really, really exciting to me and to a lot of other people in this field is that as we talk about otoprotective drugs that protect hearing, they have been very, very broadly effective in animal models with cisplatin-induced injury, with aminoglycoside-induced injury, with noise-induced injury, and even in some cases with age-related hearing loss. So the identification of some common mechanisms of cell death, common mechanisms of injury across all of these different insults and disease processes has really opened the door to the development of protective agents. So with that big picture in mind, let me introduce some of the medications that can cause hearing loss, and then we'll spend a couple of minutes talking about each of those in a little bit more detail. I think probably everybody is familiar with the big two medicines that are used to treat cancer. Cisplatin is one of the drugs that is very, very well known for causing a lot of hearing loss. Some of the other chemotherapeutics, such as carboplatin, are also ototoxic, perhaps to a bit lesser extent, but they do cause a tremendous amount of hearing loss. The aminoglycoside antibiotics, drugs like gentamicin and amikacin, streptomycin, neomycin, are also very ototoxic, causing a significant amount of hearing loss. There's some other antibiotics that have a lesser ototoxicity, that have some opportunity for either reversible or permanent hearing loss. The macrolide antibiotics, azithromycin, clarithromycin, are two of the antibiotics that are not aminoglycosides that can cause some hearing loss. And then vancomycin is another. It sounds like an aminoglycoside with the name vancomycin. It is not an aminoglycoside. It's actually a glycopeptide antibiotic. That one tends tends to be associated with a temporary hearing loss rather than a permanent hearing loss. So we tend not to hear as much about it or be as worried about monitoring for ototoxicity. I'm going to talk with you a little bit about loop diuretics tonight, which are used to treat high blood pressure and heart failure. I'm going to talk briefly about aspirin and some of the other nonsteroidal anti-inflammatory drugs such as ibuprofen and naproxen, and just give you a little bit of insight into the prevalence of hearing loss, risks for hearing loss with these different common medications. So if you're not that familiar with cisplatin, I've already described it as a chemotherapeutic. It's been around for a long time. It was identified in the 1970s. It's one of the most widely used chemotherapeutic drugs, and it's very often used in combination with other chemotherapy agents, carboplatin, oxaliplatin, vinblastine, vincristine. Um, by by coming up with these cocktail therapies or combination therapies, one of the goals is to try and reduce the side effects by using lower doses of each of the different chemotherapeutics. It can also be helpful when you have a resistance develop, and then these agents are of course also used with radiation or radiotherapy and with surgery. In terms of statistics, there is a lot of variability in the statistics on hearing loss caused by cisplatin. And in general, you'll see a number of reports describing hearing loss in about 60% of children, about 20% of adults. When you start looking at reports where they have included distortion product otoacoustic emissions to look at how the hair cells are functioning, or looking at extended high frequency data, really looking at the health of the basal cochlea, you see much, much higher rates of ototoxicity. As many to 80 to 90% of patients will experience deficits when you use some of those more sensitive measures. So that's part of the variability in the literature. And then of course the other issue is these are human studies where you have people who have different health status and people who have different cancers, different dosing regimens. They can have repeat courses of the chemotherapeutic agents, different durations of dosing. So you'll see differences in the vulnerability of the population based on all of those differences in dosing as well. One of the things that is really interesting about cisplatin, and this is a relatively new finding, this is out of Lisa Cunningham's lab at NIH in 2017. They've started looking at why cisplatin is so toxic to the cochlea. There are a lot of tissues and organs within the body that are negatively affected by cisplatin. But the cochlea seems to be pretty uniquely sensitive to injury. And so they looked at the rate of clearance of cisplatin from different tissues. And they found that in most organs, cisplatin arrives in the organ quickly. It can be detected within about an hour after the cisplatin infusions. And then it's pretty quickly eliminated over a time span that might be from a few days to a few weeks. But in the cochlea, cisplatin is it remains for very long periods of time. You can find cisplatin in the cochlea months or years after the cisplatin was administered. This has been demonstrated in both mouse models and also in human temporal bone analyses. The accumulation of cisplatin is consistently high in stria vascularis, followed by the outer hair cells, the inner hair cells, the spiral ganglion. So a number of tissues that are critically important to hearing and to cochlear function are subject to these long-term accumulations of cisplatin, which is toxic to those cells. One of the temporal bones that was evaluated in the work from Dr. Cunningham's lab, they had a small number of cochlea extracted from temporal bones where they looked at cisplatin retention. And one of the temporal bones was a pediatric temporal bone, and the cochlea from the pediatric patient was had a much higher concentration of cisplatin in it than any of the adult samples that they had. And so they have speculated that this might be one of the reasons that children appear to be so much more vulnerable to cisplatin-induced hearing loss than adults, and why there is such a higher rate of ototoxicity in children than in adults is because of differences in the accumulation and retention of cisplatin in the cochlea. Certainly the identification of cisplatin in the hair cells and in the stria has pointed to targets, therapeutic drug targets. And as we talk later tonight about otoprotection, we'll talk about the efforts and improvements that have been made with trying to protect hair cells and preserve hair cell survival despite administration of cisplatin. That work has a long history in animal models, and there's a number of groups that are now trying to move to human testing as well. Just in terms of some of the other anti-neoplastic or chemotherapeutic agents, I mentioned carboplatin earlier. It's less likely to result in ototoxicity than cisplatin, although it does still result in some hearing loss. Oxoplatin has not been as toxic to cochlear hair cells, but it does impact the auditory nerve. Vinblastine, vincristine, again, those are a lot less likely to result in hearing loss. There have been some case reports of hearing loss with those two agents, but in most cases, it occurs when those drugs are given in combination with cisplatin or with carboplatin. So it's been difficult to really tease apart a specific ototoxic effect of vinblastine or vincristine. Switching gears a little bit to aminoglycoside antibiotics. There are a couple of different mechanisms through which antibiotics have their effect. There are antibiotics that just stop bacteria growth. And so if you think about bacteria as having a lifespan, if you can stop the bacteria from growing, from reproducing, when the bacteria die, there are no new bacteria that come in and take their place. And so you essentially take the antibiotic long enough to outlast the life cycle of all of the existing bacteria. That's one of the reasons that you hear over and over again from doctors and from pharmacists that you need to take the complete course of the antibiotic, is so that you can make sure that the antibiotic is taken long enough to outlast that bacterial life cycle. There's other antibiotics that will kill the bacteria directly rather than impacting their growth and reproduction. And the aminoglycoside antibiotics are one of the antibiotics that will kill the bacteria. They are very potent in killing both gram-positive and gram-negative bacteria. And at least in the United States, they are particularly reserved for some of the more serious infections that are resistant to other bacteria because of the significant side effects, in particular the ototoxicity that is observed with the aminoglycosides. Now, this is not necessarily true around the world. There are countries where aminoglycoside antibiotics are really a first line of defense, in part because they're so effective, but also in part because they're very inexpensive. They've been around since the I think the earliest work was done in the 1940s, and slowly over a course of about 30 years, new aminoglycoside antibiotics emerged. And so they've been around a very long time. They're very inexpensive, and in countries such as South Africa, for example, they're one of the first drugs to be looked at. Streptomycin in particular is very commonly used in South Africa. In China, gentamicin is very widely used for a lot of infections. So the US is a little bit different than some of the other countries around the world. In terms of what they're used for in the United States, gentamicin is used commonly in NICU settings, particularly for prophylaxis. either with preterm infants or even with full-term infants. If an infant comes in with a fever, until they've ruled out sepsis or major infections, they may want to start an infant on gentamicin. We actually had an experience with my daughter a very long time ago when she was born, when she had a fever the day after she was discharged from the hospital, and the pediatrician sent us straight to the emergency room, and the emergency room said, we're going to start her on gentamicin right away, high dose gentamicin, until we know what's going on. And as both a new mother and somebody who knew something about gentamicin, I immediately said, no, look for another antibiotic. That's not what we're doing. But there is definitely gentamicin used in the US in those NICU and emergency settings. Tobramycin is another one that's used in the United States, particularly for respiratory infections. Patients with cystic fibrosis can have some very resistant lung infections, and tobramycin is commonly used. One of the most recent updates with tobramycin is they've developed an inhaled form. Not as much work has been done with the inhaled form to see if it has the same pattern of ototoxicity as the earlier injectable form. So that's a bit of an open question right now. And then amikacin is also used in the US, particularly in cases of drug resistance, tuberculosis and multi-drug resistant tuberculosis. Amikacin is one of the drugs that would commonly be used for those kinds of infections. Now, with the aminoglycoside antibiotics, some of them are more disruptive to the cochlea and to hearing. Others are more disruptive to balance. In general, we focus a bit more on the cochlea toxic drugs, simply because the effects on the cochlea are permanent. With the vestibular effects, there is often much more opportunity for compensatory strategies to develop, and so there are less permanent disruptions to balance than the cochlea toxic effects of hearing loss. So you'll see much more literature on the cochlea toxicity, even though there are aminoglycosides that impact the vestibular system as well. And one of the challenges for monitoring aminoglycoside-induced hearing loss is that the hearing loss can develop slowly, and it can continue to develop even weeks after the aminoglycoside has been discontinued. And so an audiogram taken at the end of the course of antibiotics is not necessarily reflective of the total amount of hearing loss that will go on to develop. And so when we see differences in rates of hearing loss with aminoglycoside antibiotics, that can be part of the story, is when the audiometric testing was done relative to when the drug delivery ended. And again, we also have differences, just as we discussed with cisplatin, related to what specific tests are done. If it's a conventional audiogram, through 8,000 hertz, the rate of ototoxicity will be much lower than if measures including otoacoustic emissions or high-frequency audiometry are used for monitoring purposes. Again, the rate of ototoxicity is much, much higher with those more sensitive measures. Your loop diuretics, ethacrynic acid, furosemide, bumetanide are the most common. Essentially what these drugs do is they pull extra fluid out of the body by acting on the kidneys. They're very common in terms of use with patients who have congestive heart failure or hypertension or pulmonary edema. They can be administered orally or by IV. IV administration will have the highest risk of ototoxicity. The other issue is if they're used in patients who have either kidney disease or liver disease. The different loop diuretics are metabolized in different organs, and so if you have kidney disease and you deliver ethacrynic acid or furosemide, which are metabolized in the kidneys, kidney disease would slow the rate at which the drug is excreted from the system, and so you'll have a longer lasting a longer lasting presence of the drug in the tissue, and the accumulation will be greater since it's clearing more slowly. And so having the drug in the system for longer periods of time is also associated with greater ototoxicity. In the case of bumetanide and torsemide, they're metabolized in the liver, and liver disease would result in slower clearance, higher accumulations of those drugs, and increased ototoxicity. Loop diuretics, when they're present at high levels, they compromise the potassium recycling in the cochlea, and they compromise the blood flow to the cochlea through their actions on potassium recycling and the strial swelling that occurs with the compromise in potassium recycling. Those effects are usually temporary, but when they're given in combination with other drugs, there can be very significant, immediate, profound hearing loss. A lot of the animal models where cochlear implants are studied, they'll use a combination of a loop diuretic and an aminoglycoside in order to cause an immediate, permanent, profound disruption to the entire hair cell population. So there's some really significant interactions with these different drugs that have toxicities in the ear. I wanted to mention quinine, chloroquine, quinidine have been in the news quite a bit. Of course, these are used to treat malaria. These chemicals are also found in tonic water, and we've known for a long time about quinine toxicity, which can include tinnitus and hearing loss, dizziness, headaches, vision changes. They're typically reversible, so we typically don't hear a lot about these deficits. In some cases, they have been permanent. Most of the literature looking at ototoxicity of these quinine derivatives have been case reports. And one of the major questions right now that is just starting to emerge in the literature is questions about increased ototoxicity, increased likelihood of permanent hearing loss for patients who have been diagnosed with COVID-19 and who take these drugs as potential therapies for the COVID-19 infection. The doses that are being used for COVID-19 are much, much higher than the doses that are being used for malaria. You can see in the review right here, the recommended dosing was 1 gram daily for 10 days compared to the malaria treatment of 1 gram daily for 3 days. There's really no systematic information right now regarding ototoxicity, but this is certainly something that a lot of people have said we have to be cognizant of and start monitoring closely so that we understand patterns of ototoxicity. All right, the last of the drugs that I wanted to mention that have some negative effects on hearing are analgesics. And certainly the amount of hearing loss that is associated with these over-the-counter painkillers is less than that of the drugs that we have been talking about so far. The effects tend to be smaller, and you see the hearing loss emerge only in these very large epidemiological analyses that I'll share with you in a moment. Because of the very, very large number of people who take over-the-counter painkillers, however, there has been an increasing amount of discussion about the potential for very significant public health impact when you have a large number of people with a lower degree of hearing loss from taking some of these over-the-counter drugs. So just so we're all using the same terminology and thinking about these drugs in the same way, the non-steroidal anti-inflammatory drugs, or NSAIDs, one of the best known is aspirin. Salicylate is the active ingredient in aspirin. Salicylate is really interesting. We're going to talk later tonight when we talk about otoprotection, about salicylate as an otoprotective agent. And so it's very dose dependent in terms of whether it will have beneficial or harmful properties. I think probably everyone is aware of the temporary tinnitus that can occur after a high dose of aspirin. Probably fewer people are familiar with the potential for hearing loss with aspirin. Some of the properties of salicylate that make it a popular choice as an analgesic are the combination of pain relief, fever reducer, anti-inflammatory, and anticoagulant properties, all of which can be reasons for taking aspirin. Some of the other NSAIDs, ibuprofen and naproxen, are two very well known NSAIDs that have received a lot of use. These are all what we would call non-selective NSAIDs because they inhibit multiple enzymes, the COX-1 and the COX-2 enzymes. There are some other NSAIDs that have a little bit different mechanism of action. They're called selective NSAIDs because they only block one of those two enzymes, only the COX-2 and not the COX-1 enzyme. You might have heard of Celebrex or Vioxx. These are popular because they have fewer GI side effects, less abdominal pain, less acid reflux, less heartburn, but they also appear to have some increased risk of heart attack and stroke. So they have not been anywhere near as widely recommended as your aspirin, ibuprofen, naproxen variants. Acetaminophen is not an NSAID, it's not an anti-inflammatory agent, but it is an over-the-counter analgesic. Obviously, it is well known to provide fever relief, pain relief. It was not originally thought to be ototoxic, although Vicodin, which is a combination of acetaminophen plus codeine, has been associated with a rapidly progressive permanent hearing loss. So some of the combinations were identified fairly early on as ototoxic, although acetaminophen itself was not. These are the analgesic epidemiological data that I referred to. And so there's a whole series of studies that have come out of Sharon Curhan and Gary Curhan's lab in Boston, and they have looked at very, very large epidemiological data sets. The health professional men's physician study has a sample of some 56,000 men who have been studied for in excess of 20 years. They have used a criteria of self-reported, professionally diagnosed hearing loss, and they find that regular use of analgesics is associated with a higher rate of self-reported, professionally diagnosed hearing loss. And it's particularly evident in men who are under 50. And essentially the interpretation has been that as you move into the 50s, the 60s, the 70s, increasing decades, age-related hearing loss starts to overwhelm any of the effects of the analgesics on hearing, and so those potentially harmful effects of analgesics are most evident in the earlier years. Use of ibuprofen or acetaminophen, but not aspirin, has been associated with an increased risk of self-reported hearing problems in women. This is data that comes from the nurses study. Again, they've been following somewhere on the order of 30,000 nurses for upwards of 20 years, and so they're able to follow things like diet, things like analgesic use, multiple health issues, self-reported hearing problems, and try and look for relationships between these different health behaviors or substance use and hearing outcomes. So either frequent use, 2 or more days per week, or a longer duration of use of some of these products, greater than 6 years compared with less than 1 year, have been associated with increased risk of hearing loss. And so this has emerged as an important and potentially modifiable contributor to hearing loss. So to wrap up the first part of what I wanted to talk to you about tonight, the drugs that can harm the ear. Again, cisplatin and the aminoglycoside antibiotics are really critically important drugs that are used in life-threatening cancer, life-threatening infection cases. The benefits of those life-saving properties outweigh the ototoxicity um, that occurs with the use of those drugs. Um, but unfortunately, they are highly ototoxic with the accumulation in the cochlea and the damaging of the hair cells, the stria, the neurons. Loop diuretics dramatically potentiate ototoxicity of other drugs. Again, generally reversible, but used in combination with some of these other toxic drugs, they can dramatically increase the risk of hearing loss. And so better understanding of the mechanisms, better understanding of how these drugs injure the hair cells, how these drugs induce the hair cells to die, um, has really opened the door for developing novel drugs that will protect the ear. And so understanding what it is that cisplatin does in the ear, what biochemical processes it initiates that result in hair cells proceeding to cell death, has opened the door for drugs that stop those biochemical events to be used to try and prevent cell death. Aminoglycoside antibiotics initiate the same set of biochemical events, and again, create opportunities for intervention. Noise injury turns on the same cascades of cell death events, opening doors for intervention. And perhaps aging. The data on aging are certainly less clear. This might be a good point if anyone has questions before we start talking about some of those targets and then drugs that act on those targets. I'm happy to field questions, or I'll keep going. All right, there will be plenty of time for questions at the end. So shifting gears, now that you know what drugs cause hearing loss and something about the hearing loss that they cause, let's talk about those drug targets that I've been introducing. So if we were to look at, this is one of my favorite pictures. My students don't really like this picture, but I think it does a beautiful job showing where the different protective agents have the opportunity to provide an intervention. And so if we were to look at something like the antioxidants, you can see that they are acting on the outer hair cells. If we look at something like the glucocorticoids, your steroids, you can see that they are primarily acting in the stria vascularis and in the lateral wall. TNF alpha gets a lot of discussion. J&K gets a lot of discussion. So all of these are molecules, either enzymes or proteins or chemicals that are created through chemical reactions that result in death of cells. And so understanding that hair cells die has opened the opportunity to look for what drugs can we use to try and protect the hair cells. Understanding something about what happens in the stria to cause it to swell and to cause cells to die has opened the opportunity to try and find drugs that will act on the stria, and so on. I'm going to show you just one more picture. And again, my students hate this one. Nobody has to memorize it, I promise, not even in my classes. But I think this is such a great image. This comes from Tom Vandewater's lab, who has done so much beautiful work on mechanisms. And it does such a nice job showing the complex cascade of events. We talk fairly simply and fairly superficially about reactive oxygen species, free radical formation, antioxidants, talking about trying to prevent those initiating events that start these pathways to cell death. But the effect of free radical formation is complicated in terms of starting these entire cascades of events. And so it's opened the the process to the development of combinations of agents. And so there's a lot of interest in using antioxidants that try and act on those earliest initiators of cell death cascades and combining them with TNF alpha blockers or with J&K blockers, or with agents that will act on the caspases themselves to try and block the caspases from having their final effects. There's two different groups of caspases, cell death initiators and cell executors. And essentially, if you intervene up to and including at the point of the cell death initiator, you have an opportunity to save the cell and prevent it from going on to die and scar. Whereas once the executioner caspases are activated, it's too late to save those cells and prevent those harmful reactions. So it's opened the door for pharmacological approaches for trying to protect the ear. There was a fantastic summit that was held in 2018 that was hosted by Action on Hearing Loss, which is a charitable organization in the United Kingdom, which is a group that it provides services for people with hearing loss, advocates for people with hearing loss, and provides funding for research that is intended to prevent hearing loss or restore hearing. And we don't have time to go through all of the preclinical data tonight, so I'm going to make three broad statements that I hope you'll trust me on. And you can, of course, feel free to reach out to me for some recommendations on reading or more information. But the first thing I want to share with you is that when we think about drugs that act on the pathways that I have just shown you in the last two slides, there is a tremendous extensive literature that documents in vitro protection of hair cells using different drugs that act on those pathways. And so if you put hair cells into a dish and you put cisplatin in the dish and you put a protective agent that will block the pathways we've just been looking at in the dish with the cisplatin and the hair cells, you can protect the hair cells so that they don't die from the exposure to cisplatin in the dish. And that's a really common way of doing some initial screening, because it is a very quick and easy scan to see if cells are dying or being protected. When drugs are successful in those in vitro models, either with cell lines or with hair cells themselves, one of the next steps is to move to in vivo models. And again, there's a very extensive literature documenting protection in vivo in living animal models, showing protection of hair cells using these different drug agents that act on the pathways that we've just been talking about. And so you can take an animal and treat the animal with cisplatin or treat the animal with an aminoglycoside, and then in parallel to the cisplatin or to the aminoglycoside, deliver a protective agent, and you will be able to preserve the hair cells so that they don't go on to die in the same way that they do in an animal that did not get the protective agent. Of course, we're not just interested in did we save the hair cells from dying? We're interested in, do they actually work, and did we actually protect hearing? And so there's a very extensive literature documenting, say, in vivo protection of hearing sensitivity using these otoprotective drugs in combination with various chemicals. So I have just summarized probably 35 years of work from 20 labs around the world in summarizing that information for you. In terms of what are some of those agents, I'm going to just quickly show you some of the the agents that have been successful with different kinds of insults and injuries, because I want to highlight some of the similarities across these different models. And so if we were to look at cisplatin ototoxicity, you would see a tremendous literature looking at dimethionine. You would see information collected using epsilon either by itself or in combination with another antioxidant called allopurinol. N-acetylcysteine has been investigated. Salicylate, remember this is our active agent in aspirin, has been protective against cisplatin ototoxicity. Vitamin E, vitamin C. Amifostine has been successful in animal models. That's a drug that did actually move into human testing, and there were 4 completed trials. There was not a significant benefit with the meta-analysis of those trials. So there's a few questions about clinical viability of amifostine as a protective agent. And then sodium thiosulfate. I'll show you some data that was collected in a clinical trial from sodium thiosulfate. It's important to introduce at this point that the way that sodium thiosulfate acts is it is an antioxidant, but it also directly neutralizes cisplatin. And so this is one of the major challenges for using sodium thiosulfate to try and protect the ear, because a major risk is that you can't inactivate the effects of cisplatin on the tumor. So you have to provide enough of a time buffer for cisplatin to get to the tumor and to have anti-tumor effects, or to keep the sodium thiosulfate localized to the ear so that it doesn't compromise the effects of cisplatin on the tumor. Because if all you do is neutralize cisplatin so that it can't affect the ear, you're also neutralizing cisplatin so that it can't affect the tumor. So this is one of the reasons that efforts to try and move otoprotection against cisplatin and against aminoglycosides into human testing has been so much slower than efforts to study otoprotection in noise injury models, because of those concerns about compromising the important life-saving benefits of the cisplatin or the aminoglycoside agent. Looking at aminoglycosides, you'll see some very similar otoprotective agents. Dimethionine appears again, epsilon appears again, sodium salicylate appears again, N-acetylcysteine appears again, vitamin E appears again, vitamin C appears again. Coenzyme Q and ginkgo biloba have both been studied for potential benefits against aminoglycoside ototoxicity. We've done a little bit of work with a combination of vitamins, beta carotene, vitamin C, vitamin E, and magnesium. And then, of course, noise models. Again, you'll see some similar overlap. I have put a box around the dietary supplement and epsilon simply because those are the 2 agents that we've studied in clinical trials previously, and the patent protection that I mentioned in the beginning is for the combination of vitamins and magnesium. Dimethionine, you can see, has been very, very well studied in noise models, N-acetylcysteine, salicylate, and I included a couple of additional antioxidants here, the U74-9 or 389F, the SOD polyethylene glycol. Those are agents that go all the way back into the early 90s, and so we're going on just about 30 years now since the first efforts to try and use antioxidants to try and prevent ototoxicity due to noise exposure, followed by all of the work with cisplatin and with aminoglycosides. One of the things that is so exciting about the 3 slides I've just shown you every single one of these studies has shown some benefit of these antioxidants in preventing either the cisplatin-induced injury, the aminoglycoside-induced injury, or the noise-induced injury. And to have that kind of consistent positive benefit across a wide range of antioxidants that are targeting shared mechanisms is really, really encouraging in terms of the potential for translation to humans. It's concerning how to try and evaluate what the best agent will be, however, and let me spend a little bit of time talking about that next. So there's a big problem trying to compare drugs across data sets. And the major challenge is, if you think about how each of these drugs have been developed, every lab that's working with a particular otoprotective drug has a preferred animal model. Some of the drugs are tested in guinea pigs, some are tested in rats, some are tested in chinchillas, some are tested in mice. Each of those rodent models, while they're all rodents, they have very different digestive systems, and so the drugs are metabolized at very different rates. They also have very different metabolism in general. A mouse has a much, much higher metabolism than a larger rodent. You need to give a mouse about 5 times as much of a drug in order to have the same effects, as the drug given in another larger species. Another major challenge is that each of these rodent species has a different vulnerability to either aminoglycosides or cisplatin or noise. And even if you have 2 investigators who are both working with guinea pigs for prevention of noise-induced hearing loss, you'll find that they'll potentially use very different noise exposures, resulting in different amounts of hearing loss in the control animals. And so if you have one injury model that causes a very large, profound hearing loss, and another injury model that causes a much smaller degree of hearing loss, and then you have otoprotective agents, and one otoprotective agent completely prevents the small change in hearing, and a different otoprotective agent partially prevents the large change in hearing, trying to identify which one is more effective is impossible, given that you don't know if the drug that completely prevented the small change would completely prevent a large change. You don't know if the drug that had some benefit against the large insult would have completely prevented the smaller insult. So I'm always very, very careful to tell people that the way that the preclinical data have been collected make it really impossible to try and draw conclusions about what the most effective antioxidant is going to be, particularly with respect to humans. There's a host of other differences across studies. Some investigations will initiate the protective treatment several days before the injury. Others will start it at the time of injury or even after the injury. The duration of time over which the protective agent is administered can vary. Some will provide the agent just during the time of insult. Others will continue to provide the protective agent for multiple days or even multiple weeks after the exposure to the insult. The method of administration, whether it's oral or injected, varies. How many times per day the drug is given varies. And perhaps most concerning of all is there has been almost no effort to look at dose response curves. When we talk about dose response curves, we're talking about looking for the most effective dose of a drug so that you can be sure you're comparing the best dose of one drug to the best dose of another drug. And what has actually happened is labs have picked an agent that they want to investigate, and they've done some preliminary work where they found benefit, and then rather than systematically manipulating the dose of the drug in order to make sure they're using the most effective dose, they simply continue to do all of their work using the dose that they identified as beneficial in their initial studies. And so we have no idea if we're even comparing the best dose of one drug to the best dose of another drug within the animal models. So it's really unfortunate that there has not been better consensus in the way that otoprotection has been investigated. So going back to my initial statement, it is tremendously exciting that we have seen so many agents that have provided at least some protection against each of these different injuries and insults, but we really have no idea from the animal data which of those are going to be the best agents to pursue in humans. For better or for worse, this has not stopped efforts to develop multiple drugs for use in humans. And so if we look at some recent reviews, this is a review by Tanisha Hamill and Kathy Campbell, which was published just a year ago in 2018. And they went through the literature and they identified interventions that are being tested in humans. And so you can see the drugs listed on the left. You can see the different targets that they've been tested for in animal models where they have shown some benefit in those animal models. And then in the third column, the intervention column, there is a clinical trial number and the indication that the drug is being tested for in humans. And so you can see that there's a number of drugs that are being tested against cisplatin ototoxicity, and it is tremendously exciting to think that at some point one or more of the drugs on this list that are being tested to try and prevent cisplatin ototoxicity could actually be shown to have some benefit and make it through the FDA review process and ultimately be approved for use in human patients who have to take cisplatin for life-saving purposes. You can see there's very, very little work on aminoglycoside-induced hearing loss. Epsilon, there is a clinical trial that's looking at tobramycin-induced hearing loss. This is a study that's being done in patients with cystic fibrosis who are taking tobramycin for respiratory infections. The vast majority of the studies listed that were found in this review are studies looking at protection against cisplatin ototoxicity. If you're not familiar with clinicaltrials.gov, this is a great resource that you can consider looking at in the future. Clinicaltrials.gov is organized under the NIH offices, and any of the clinical trials that are funded by NIH have to be listed on clinicaltrials.gov. The FDA also requires that clinical trials be posted in a clinical trial registry, and clinicaltrials.gov is the primary registry within the United States. And so most of your drugs that are being tested under IND, investigational new drug applications, or NDA, new drug application trials are going to be listed in clinicaltrials.gov. There are also some European registries and an Australian registries, so this is not the only source of information worldwide, but certainly most of the trials within the United States will be listed within clinicaltrials.gov. And what this does is it lets you enter a particular health indication that you're interested in, or if you know of a drug, you could enter the drug name. There's opportunities for different searches, but it will tell you, it will list all of the clinical trials that are being done for a particular indication. So you can enter tinnitus and see all of the clinical trials that are being done to try and ameliorate tinnitus. You can enter cisplatin and see all of the studies that are being done to try and reduce cisplatin-induced hearing loss. You could enter aminoglycosides as search terms, or you can enter noise as a search term. And this is another review. This is a review that I published last year, which rather than looking at the cisplatin and aminoglycoside trials is looking at studies that are going on in humans to study protection against noise-induced hearing loss. And so again, you can see that there's a study ID, a clinical trial number. You can see the titles of those studies. You can see what intervention is being assessed. You can see the company that is sponsoring the trial. And then you can see also the status of the trial. And one of the nice things about clinicaltrials.gov is there's a requirement that results be posted. And so one of the challenges with drug research is that when a drug is not successful, it can be difficult to publish negative results in some of your journals. And it can also be of low appeal, of limited interest to the companies that are developing those drugs to publicize the drug failures. Of course, this also means that you can end up where you have multiple groups retesting the same agents, not knowing that someone else has already tested it and found that it was ineffective. So clinicaltrials.gov has requirements for results of studies to be posted regardless of whether the data go through a peer review process or not. There is a very, very extensive vetting of the posting of the results by the clinicaltrials.gov team. I can tell you from personal experience. So results can be slow to make it into that listing, but eventually they do, which is really helpful, and I think a major advantage that this has become a requirement. If you're interested in learning more about the drugs that are being developed for potential use in humans, there is a fantastic review by Ann Schilder and her team that came out earlier this year. And they did a search, the actual search process was done in June 2018, and they looked for companies that are working in the field of therapeutics for the inner ear and central hearing disorders. So it's not your traditional PubMed type search, but rather a search of all of the biotech companies, biotech statements, biotech websites, looking for companies that have clinical or preclinical programs with agents that they hold the rights to develop. And I've captured their results here for you. If you look at otoprotection, you can see that there are 24 companies that are pursuing 37 different therapeutic programs, some of them related to ototoxicity, drug-induced hearing loss, some of them related to noise injury, some related to aging, some related to sudden hearing loss, some related to loss of residual hearing during CI insertion, cochlear implant insertion. Tremendously exciting, though, to see that kind of investment. If you think about where we were 10 years ago, there were maybe 3 companies that were starting to develop agents, and they were small companies with very limited funds. Now we're seeing very large venture capital investments, very large pharmaceutical firms. So it's a completely different landscape than what we had 10 years ago. You can see that there also are companies that are working on regeneration therapies, 15 companies working on 13 therapeutic programs. Of course, a lot of these are in early stages. Preclinical would be in your animal research phase. Phase 1 is your human safety. And then phase 2 is when you first begin looking for efficacy of those agents. There are also efforts to look at tinnitus reduction, really limited efforts to look for drugs that will act on central hearing disorders, drugs that will act on the brain rather than on the periphery, and then a few programs that are working on balance programs. In terms of human data that have been collected, I want to spend the last portion of this talk just speaking with you about some of the work that has been done in humans so far. Cisplatin, I mentioned, is one of the agents that there has been some work done, and I wanted to show you a little bit of data from an otoprotection study that was done in humans using sodium thiosulfate. And this is the drug that I mentioned inactivates the sodium thiosulfate, and so it requires some care in the way that it is administered. This was a large study that enrolled children ages 1 to 18 with newly diagnosed cancer, and they were randomized to receive either cisplatin plus observation or cisplatin plus sodium thiosulfate. And the sodium thiosulfate was given 6 hours after the cisplatin dose to try and reduce the potential for compromising the anti-neoplastic benefits of the cisplatin. And their primary endpoint was incidence of hearing loss 4 weeks after the final cisplatin dose. And the way that they measured hearing was using ASHA's standard definition of a significant threshold shift. So they were looking for the rate at which large changes in hearing occurred. If you're not familiar with ASHA's definition of an STS, a 20 dB change at any one frequency, a 10 dB change at any 2 adjacent test frequencies, or loss of response at 3 consecutive frequencies where a response was obtained at baseline would be the criteria for meeting ASHA STS. And so from an audiometric perspective, the results were very, very encouraging. You can see that there was a rate of ASHA STS in about 29% of the children who received cisplatin plus sodium thiosulfate versus 56% of children who received only the cisplatin, which was a statistically significant result. There was a much reduced rate of hearing loss, reduced odds of developing hearing loss in the children who received sodium thiosulfate prior to cisplatin, or after, 6 hours after cisplatin. They also followed the children 3 years later to look at the overall outcomes, and this is where some of those cautions come into play. If you look at the 3-year event-free survival, they found that that survival was 54% in the sodium thiosulfate group and 64% in the control group. Overall survival was 70% in the sodium thiosulfate group and 87% in the control group. So they had a much poorer long-term survival and also a much poorer prognosis in terms of event-free survival, meaning did the cancer return. They did some subgroup analyses to look at different types of cancer, and they found that patients who had a disseminated metastatic disease who were treated with sodium thiosulfate plus cisplatin had lower event-free survival and lower overall survival rates than the cisplatin-only patients. Patients who had a localized disease, they didn't see any differences in survival. So these results are a really important cautionary tale for work on ototoxicity prevention when you have the potential for interactions between your anti-neoplastic drug, your chemotherapeutic drug, and your otoprotective agent. A little bit of data on aminoglycosides from humans. This is a study that was done in China, and these were patients who were being treated for various infections, and they required gentamicin for for treatment of their infections. And so they were randomized into two groups, either to receive gentamicin plus aspirin or to receive gentamicin alone. The definition of hearing loss was a bit different in this group. They defined hearing loss as a 15 dB or more shift at the higher frequencies at 6 and 8 kilohertz. And they found that the incidence of hearing loss using that criteria was reduced from about 13% in the control group that did not get aspirin to about 3% in the treated group that received aspirin in addition to gentamicin. The primary adverse event was gastric upset, so there was a lot of gastric bleeding and a lot of gastric distress with the high doses of aspirin, and so that is one challenge for trying to move forwards with a broader use of aspirin in otoprotective scenarios. This is another study looking at protection against aminoglycoside ototoxicity, and this is amikacin ototoxicity. There's a lot of information on this figure, so let me just fill you in on the key highlights. So if you look at that top panel, what you are looking at is an early test time, 1 week after the amikacin or amikacin combination was started. PTA1 is low frequencies, PTA2 is high frequencies. So PTA1 is going to be an average of all of your frequencies from 250 hertz to 8,000 hertz. PTA2 is going to range from 10 to 16 kilohertz averaged across all of those frequencies. And so you can see that there is considerably less hearing loss, less hearing shift in the patients who received N-acetylcysteine in addition to amikacin at that 1 week test time. If you look down at the bottom panel, you will see that this is now 1 month after the amikacin administration, and you can see that the controls who did not receive N-acetylcysteine have about 15 to 20 dB of hearing loss at both the low and the high frequencies, whereas the group that received N-acetylcysteine developed very, very little hearing loss from the amikacin regimen. In terms of noise, there is quite a bit more that has been done and has been published. And I just want to briefly introduce one of the challenges for noise research, and that's that most of the preclinical studies have looked at reduction of permanent threshold shift, which is what we're largely ultimately interested in. But most of the human trials that have been done to date have looked at prevention of temporary threshold shift. And so if you think about why you might use a TTS model to study otoprotection against noise injury in humans, part of it is logistical issues. And so if the TTS studies are much, much shorter in duration, if you can look at preventing changes in hearing that occur with a single short exposure instead of trying to follow someone for multiple years of slow development of occupational noise injury. Shorter studies, significantly less expensive to do those short-term studies with a short period of drug administration. You are much less likely to have your subjects drop out if you're only asking them to participate for a week instead of participate for several years. And participants are also assumed to be safer because there is no permanent damage that's expected from participation in those studies. There has been recent interest in TTS studies as having significant value in and of themselves, simply because of the tremendous importance of preserving communication, audibility, and understanding of sound in noisy, chaotic battlefield situations where it can be very important to distinguish the difference between words like attack and fall back. So preserving hearing in those acute situations is critical. The way that I have thought about the role of TTS studies sort of more broadly is that they provide a very important proof of concept, that they show the drug is available in the cochlea at a concentration that is sufficient to protect the ear against a small transient injury. Which clearly the drug would have to be present in the cochlea in order to prevent those longer-term injuries as well. And most, if not all, of the agents that have reduced TTS in animals have also reduced PTS in animals. So there's been a very, very good correlation between protective benefits against TTS and PTS. Of course, confirmatory data in PTS trials will be needed in order to be able to claim protection against permanent hearing loss. But with that, let me just quickly show you some data from temporary threshold shift studies, and then we'll wrap up and take some questions. We've done a series of studies. We've looked at prevention of temporary threshold shift after weapons training with the Swedish military population. We have looked for prevention of TTS after use of music players, and we are currently in the process of launching 2 new studies which will look at prevention of noise-induced hearing loss that occurs with surgical skull-based drilling noise. That will also have some vibration components to it, of course, and then prevention of threshold shift after weapons training in a population of safety officers. So just to introduce the paradigm for the Swedish soldier study, they were required as part of their military training to participate in what they referred to as urban combat exercises. They essentially went into a concrete bunker and participated in training with machine guns, 20 shots per round, to rounds, total of 40 shots of the machine gun inside the concrete bunker. Very high sound levels, even underneath their hearing protector, and they had a history of threshold shift of hearing loss after participation in these training exercises. This is what we call a double-blind crossover study, and so the soldiers went through a consent process, a screening process, and they were given a bottle that contained either a placebo or a micronutrient combination. They didn't know which they were getting. We didn't know which one we were giving them. They did the training exercises. We monitored changes in their hearing. There was a period of about 3 months. And then they came back for additional training. We repeated their baseline tests. They got a second bottle of medication. If they had had nutrients the first time, they had placebo the second time. If they'd had placebo the first time, they got nutrients the second time. They repeated the training exercises, and we measured their hearing again. Now, this was a good news, bad news sort of situation. We did not have any reliable temporary threshold shift in the soldiers who participated in these training exercises during either the placebo phase or the nutrient phase for each of the participants. And, you know, one possibility, the military is constantly upgrading their hearing protectors, so it's possible that they had more effective hearing protection than in previous scenarios where they had observed hearing loss. It is also possible that the subjects were simply using their hearing protection much more carefully, that because they were participating in a study on hearing, they were very aware of their hearing, they were cautious with their hearing protection. So the good news is the hearing protection protected their hearing. There was no hearing loss in anyone. The bad news is it was essentially a failed clinical trial, because you can't measure protection if you don't have any deficits. But we were able to confirm at least that blood levels of the nutrients increased when they were in the nutrient condition, but not in the placebo condition. So this led us to do some laboratory-based studies where we didn't have to worry about how well someone was wearing their hearing protection, that we would have a small but consistent change in hearing across participants. And you can see that the studies that we did in the lab, participants only come in once, and so they either get a placebo or they get the active agent. They listen to music on an iPod, and then their hearing is tested multiple times over the next several hours after listening to the music. And these are data from two studies that I wanted to share with you. The study on the left is with the nutrient combination. You can see that it had absolutely no benefit in reducing the amount of threshold shift after listening to music. There was about a 4 dB change in hearing at 4 kilohertz in both the group receiving the placebo and the group receiving the supplement. The study that was sponsored by Sound Pharmaceuticals using Epsilon had a very different pattern of results. The black line is the placebo condition, and you can see that the placebo-treated subjects look very, very similar to both groups in the nutrient study. What was different is a dose-dependent protection. So the 400 milligram dose of Epsilon was very, very effective in preventing temporary threshold shift in the clinical trial that we completed with Sound Pharmaceuticals. So they are working to initiate a multi-site study that will hopefully be able to repeat those results. There's a couple of other studies that have been done trying to look at PTS. There's a study that was done at Camp Pendleton out in California, where they were looking for protection against changes in hearing that develop with 16 days of basic training. So these are subjects who are exposed to a lot of rifle fire, as well as some simulated explosions. Depending on the outcome measure, they detected either no benefit or some benefit. So if they looked at ASHA STS, as was measured in the sodium thiosulfate study that we looked at earlier, they did not see a difference in the placebo group and the N-acetylcysteine group. If they looked at smaller changes in hearing, an increase of 15 dB at any test frequency, they did find a reduction in the rate at which hearing loss was observed in the N-acetylcysteine treated participants looking in the trigger hand ear. And then the last study that I'll show you is the prevention of STS in service members. This is another military study. It was done at Fort Jackson with the drill sergeant instructor trainer population. Again, it's a weapons training study with a large number of weapon discharges. Unfortunately, they had a higher rate of screening failures than they expected and a higher rate of dropouts than they expected. There was a lot of GI disturbance in the people who were participating in the study. The incidence of threshold shift was lower than they expected, and the study was terminated early, so they did not have the statistical power that they hoped for, and they did not see compelling evidence of benefit with the demethionine in that study. You might be surprised, given everything I've told you about nothing having been approved by the FDA and lots of work by different companies to test drugs. If you look on the internet, these are all products that are currently available to buy over the counter right now, none of which have been approved by the FDA for prevention of noise-induced hearing loss or drug-induced hearing loss or tinnitus amelioration. So I do encourage people to be really cautious about assuming that these agents work just because they're available over the counter. If you were to look at WebMD to see what they have to say about the active agents that are available in those over-the-counter products. You can see insufficient evidence, insufficient evidence, insufficient evidence, insufficient evidence, insufficient evidence, pretty much the whole way down. So please do be very cautious about things that you see simply sold over the counter. And also, as a reminder, the FDA does not require that dietary supplements be demonstrated to be either safe or effective prior to marketing. And so over-the-counter product sales don't assure that a supplement is going to be safe either at all doses or for all people. And there's a number of over-the-counter products that can cause serious or life-threatening side effects. Some of you might be familiar with the effect of high doses of magnesium, which has laxative effects. High doses of vitamin E shouldn't be taken prior to surgery or by patients on blood thinners because of the blood thinning effects of vitamin E. Beta carotene, there's been an increased risk of lung cancer in people who take high doses of beta carotene, in smokers and workers with exposure to asbestos. Vitamin A overdose causes birth defects in pregnant women. Vitamin B6 overdose can cause peripheral neuropathy, numbness in fingers and so on. So please do be cautious with over-the-counter supplements, even if they're products that you assume are likely to be safe, and certainly consult with a doctor or pharmacist about safety if you're taking prescription medication for any health issues. The last question I wanted to address, and I promise I'll wrap it up because I've been monopolizing your attention for quite some time, not 30 hours, but quite some time, and I appreciate your attention, is what about antioxidants that are obtained via healthy diets? If you look at the popular press, you'll see that there are all kinds of images talking about food as medicine, and I just grabbed a couple of images to highlight that messaging that is appearing in the press. And there's actually a very large and very mixed literature on this topic, which we will not go into tonight. I'll just show you one slide with one data set which looks at interactions between noise exposure, hearing, and dietary quality. And so if you look at the filled circles, those are people who have low noise exposure. This comes from a very large data set, the NHANES data set collected by the CDC, which recruits about 20,000 people per cycle. And if you look within the group of people who have low noise exposure, if you look at their high frequency hearing threshold, so this is from 3,000 to 8,000 hertz, and you look at their dietary quality, you can see that the impact of dietary quality is not very large. There's about a dB of difference with the better dietary quality subset of the population having slightly better hearing at the high frequencies than the bottom 40%. This is after adjusting for age and socioeconomic status and smoking and cardiovascular disease and a host of other issues that would potentially explain differences in hearing. If you look, however, at the subset of the population that has more noise exposure, people who have occupational noise exposure or military experience or civilians who use firearms, there's a broad group of people in that category. You can see that there is a much more robust difference in their hearing at the higher frequencies that are affected by noise exposure when you compare those with better diets and those with poorer diets. Within the better diet group, the top 60% of dietary quality group, you can see that having significant noise exposure had some effect on hearing. But if you compare the effective noise on hearing, more noise versus less noise, within the subset that had poor diets, you can see that there's a much more dramatic effect of the increasing noise exposure on hearing. So I do recommend healthy diets as a potentially beneficial hearing health strategy. Certainly it's not going to hurt anybody to be eating a healthy diet with lots of fruits and vegetables, assuming that we're not in the middle of one of the lettuce E. coli outbreaks. So with that, to briefly summarize everything we've talked about tonight, it is very, very clear that oxidative stress drives noise-induced hearing loss, cisplatin-induced hearing loss, aminoglycoside, antibiotic-induced hearing loss. These aren't the only cell death events initiated, but they account for a major portion of cell death, and antioxidants have been extremely effective in preventing or at least reducing hearing loss after all of these injuries. It may be that the incomplete protection that is often observed is due to some of the other biochemical events that are separate from the oxidative stress pathways. It may also be simply that there has been far too little effort and attempt to try and come up with the most effective doses. Success in humans has been mixed, as we've looked at tonight, and nothing is approved by the FDA at this time. But certainly pharmaceutical industry and venture capital are making tremendous investments into developing pharmaceutical agents for hearing loss prevention purposes for these different indications. Trial design is a real challenge, including safety issues related to drug-drug interactions and ethics associated with making sure you protect against the effects of noise using established HPD solutions, when you're talking about preventing PTS. The development of novel protective drugs has the potential to benefit a tremendous variety of sound-exposed populations, potentially including musicians, sound engineers, other performing artists. There's an article by Frank Wardinger, Heather Maliak, and Corey Portnoff that just came out at the end of 2019 in JASA, which talks about the potential application of these kinds of agents to performing artists. And again, healthy eating is good advice regardless of whether it provides benefit or not. So for those of you who are interested in more information about anything we've talked about tonight, there is a special issue of JASA that came out in November, which has 32 manuscripts that are all related to drug development for otoprotection, talking about animal models, noise exposure models, at-risk human populations, and all of the different factors that influence individual risk, individual vulnerability. There are a number of other open access special issues. The DoD has been really prolific in putting together special issues on pharmaceutical interventions. The first 4 series of papers listed on this slide are all open access, freely available. The last one on this list is a seminars and hearing issue specifically on pharmacology and ototoxicity, but not available open access. This one was organized by a different group other than the DoD. So with that, I'm going to close and answer any questions that you have. I put up these pictures of UT Dallas and the Collier Center just to remind myself what it looks like, since I haven't actually been there since the middle of March. But any of you who are in Dallas once things open, please feel free to reach out to me at any point if you would like to see our campus and our facilities. So thank you, and with that, I'll take any questions.